system. There's a lot to the nervous system. Obviously, it's incredibly complex. I'm going to spend roughly equal amounts of time talking about the physiology and then a bit about the anatomy. When you take Dr. Farkas's neuroanatomy course, you're going to go into more detail than I go into here. But this class is definitely going to help prepare you for that class too. In terms of the neurophysiology, we're not going to go into a tremendous amount of detail, but there's still a lot to know. It's a very complex system. Obviously, the function of it is tremendously complex, and you can take, year-long. When I took neurobiology, it was a 2-semester course, 4 credits each semester. So, and that was a long time ago. We know a lot more now than we did back then. The nervous system, I'm going to talk about a couple basic divisions to give you sort of a forest view, if you will. We divide it into the central nervous system and the peripheral nervous system. The central nervous system, I hope you know already, brain and spinal cord. That's one of the most basic things you learn about the nervous system. So the brain and the spinal cord together are the central nervous system. The peripheral nervous system is everything outside of that. So that's going to include things like nerves to the face. We're going to have ganglia all over the body. Ganglia are clusters of neuron cell bodies. Nerves to upper, lower limbs, everywhere else. Okay, so we're going to divide again the central nervous system and the peripheral nervous system. Now, in addition to the anatomy, functionally, the peripheral nervous system is going to have 2 major divisions. We can talk about the autonomic nervous system and the somatic peripheral nervous system. The autonomic nervous system is further divided into the sympathetic and the parasympathetic. The autonomic nervous system innervates things that are automatic. You don't have to think about them. And they don't typically enter into consciousness. You don't know, you don't perceive directly when your sympathetic nervous system is activated. Right? There's some indirect signs, okay, but you don't directly measure that. The somatic nervous system is sensation. Okay, so we're going to deal with sensation through the somatic nervous system. When we talked about the senses, much of that was mediated by the somatic division of the peripheral nervous system. Good place to start. It'll make remembering some of the anatomy and the function of these things easier if you remember these basic divisions. All right, and I'll go over those in a couple slides. As far as function goes, here's a few. One is sensory input. You can't respond to anything, either internally or externally, unless you have sensory input. Everything we do, you require sensory input. And if you have challenges with those sensory inputs, again, it can lead to great dysregulation and dysfunction. We're also going to integrate things. In order to decide what to do, essentially, we got to take a bunch of different and sometimes very disparate pieces of information, put them together, figure out what they mean, and come up with one answer. And we'll talk a little bit what this means at the neural level and a little bit at the organismal level. It controls muscles and glands, both voluntary, skeletal, and smooth muscle. In terms of the glands, normally we don't think about the nervous system controlling gland secretion, but as you learned in digestion, right, parasympathetic activity stimulates the salivary glands, for example. When we're talking about mental activity, here we're talking about cognition. A lot of this is going to be psychology. We'll talk a bit about that. It's also key for maintaining homeostasis. So for homeostasis, right, that's where your body tries to maintain specific values. So body temperature, for example, if body temperature starts to go up, we're going to have correctional mechanisms that kick in to try to bring it down. Now, the places that detect body temperature and compare it with our set point, those are going to be located in the nervous system. And we'll talk about some specifics with that. Now, as I mentioned, we can talk about two divisions of the nervous system, the CNS and the PNS. Very straightforward as to what they are. As for their location, right, central nervous system is going to be the brain and the spinal cord. Obviously here, human brain, human spinal cord. The peripheral nervous system is going to be everything else. And amongst the structures of the peripheral nervous system, we will have the 12 cranial nerves as well as the spinal nerves. You should remember these numbers. There's 12 pairs of cranial nerves, 31 spinal nerves. And we'll get a little bit into the detail about the structure of the spinal nerves in a bit. Now first, we'll start with the most basic unit of the nervous system, and that's going to be the cell. When we talk about a cell, almost exclusively our minds are going to jump to neurons. Right? Neurons are obviously a really important part of the nervous system, but they're not the only cells that are important. In addition to having neurons, we have to have support cells. You can think of a neuron as a fighter jet in a way. You know, that's the thing that kind of gets all the glory. Everyone thinks of it. But to keep that fighter jet in the air, we need a huge crew of people. Often it takes longer to maintain it than it does to fly it. So there's some jets that are 30 to 40 maintenance hours per hour of flight. Okay? Neurons are kind of like that. Neurons are incredibly picky. They're very tough to keep alive. The glia cells, that's those support cells, the glial cells are going to help do that. When we think of pathology, problems with the nervous system, we focus on neurons. A big part of that has been our ignorance. Now, and I think you'll see in the next 5 or 10 years, we're going to spend a lot more time talking about disorders of the glial cells. That when these glial cells don't work right, the neurons don't work right. And yeah, we can focus on the problems with the neurons. That's going to tell us something. But the way to fix it isn't to do anything to the neuron. The way to fix that would be to fix the glial cell, right? Okay. So when we're talking about cells of the nervous system, we're primarily going to focus on neurons, but you also need to remember glia are there and that they're very important. In terms of sheer numbers 10 to 1 glia to neurons. So you have far more glial cells than you've got neurons. Now, let's start with the neuron though. Each cell type in the central nervous system has some very distinctive characteristics. The neuron is often called a nerve cell. Neurons require a lot of oxygen and a lot of glucose. One thing that is different about a neuron, say compared to a muscle cell, is that we have very limited storage. Okay? So if a neuron is cut off from its blood supply, it doesn't get oxygen and it's not going to get glucose. Within minutes, that neuron is going to stop functioning properly. And within 15 to 20 minutes, that neuron will die because of a lack of nutrients. So they basically starve to death and they die. You can't store this with muscle, right? If you remember the Phil's lab where we took the frog gastrocnemius or frog cardiac muscle. What you saw in those simulations, you can really do. When I was in physiology lab, that's what we did. We took a frog heart out, we'd leave it out for hours, 3-4 hours, and it would still work. You could never, ever, ever do that with a neuron. That's because the muscle cells store a lot of energy. The capacity to generate and create energy is much stronger. Neurons have to get it immediately from the blood. That's why if blood supply is cut off to a muscle, you got a while before you really have to worry about necrosis, about it dying. If you're having a stroke, which is essentially interruption of blood supply, you have minutes before there's permanent damage. So the idea is a neuron is going to receive information. It's going to try to process that. And then it's going to produce a response. Now, I'm going to simplify for this class. What I'm going to tell you is not strictly true. There are exceptions. But in general, we're going to get input from other neurons at the dendrites. So the dendrites are going to collect information. This processing is going to be largely passive physics, right? And you'll see what I mean by that. It's just going to be physical properties. The cell doesn't need to actively do anything. Produce a response is going to be the action potential. Now with the action potential, this signal is all or none, yes or no, pass, fail. Either you have an action potential or you don't. There's no such thing as a bigger or a smaller action potential. You have it or you don't. This is a true dichotomy. Okay? So what the neuron is going to do is collect information from other cells. We're going to have integration, that's the processing. And then the cell is going to produce a response. Now that response to all those signals, if it's strongly excitatory, we're going to want to tell someone. And the way the neuron tells another cell is by producing an action potential. Again, yes or no. Either we have it or we don't. If all that information coming in is not very exciting and we don't want to tell another cell, then we don't produce an action potential. When we look at the cell, we'll talk about specializations in both structure and function for receiving information and producing the response. We're not going to talk too much about the processing. Again, mostly that's physics, mathematical in many ways and beyond the scope of this class. So I mentioned that dendrites are going to receive information from other cells. Okay. So they can receive it from other neurons. Sometimes there's receptors. We talked about the cutaneous receptors, for example. The receptors are going to essentially send information to the dendrites. Okay? Now this is not the only place on the cell where we can collect information. All right, for our simplified purposes right now, I want you to think of dendrites as receiving inputs. It turns out that we can get information going to the cell body, to the axon, to the terminals. I'll talk about all of those later. For right now though, think about the dendrite as the place on the neuron that's going to receive information. Now we're going to have a cell body. The cell body has a nucleus, so this has the instructions for producing the proteins that are going to allow that cell to function. So when we look at things like protein synthesis, most of that occurs in the cell body. It's not the only place it happens, but that's where most of it happens. The cell body is also going to be important for that process of integration. Right? So when we say processes, really we're talking about integration. And I'll talk a little bit more about that in a minute. Right now you don't have the tools to understand what I mean by passive integration. And then finally we've got the axon. The axon is going to transmit a signal to another cell. And it can do this to a variety of different cells, to glands, to muscles, to an organ system, or even other neurons. Right? And when we think about this transmission, in terms of the nervous system, mostly we're going to be talking about neurons communicating with other neurons. In terms of physiology in the periphery, though, when we're talking about movement, release of hormones in response to sympathetic activation, and then we're going to think about these things. A neuron has polarity. So again, with polarity, that means that one side is different than the other side. You're used to that with a battery. You'll have a positive and a negative pole. Same with magnets, north, south. Right? So in a neuron, very simply, the dendrite is going to receive information and then pass that information through the cell body, down the axon to the next. Okay, so we go from receiving to sending information. Now, when we look at the structure of a neuron, you've had this way back in earlier chapter. I hope you remember it. If not, refresh your memory. When we look at the structure of the neuron, again, we're going to have the cell body. Coming off the cell body, we're going to have lots of different structures. Each one of those is called a process. So a neuron's going to have processes coming off of it. Now when we look at the processes, depending on what stain you're using, you may not be able to tell which are dendrites and which are axons. A neuron has one axon though. No one has found a neuron that has more than one axon. Okay? So again, one axon, that's it. Everything else coming off the cell is going to be a dendrite. So each cell is going to have many processes. You know one's an axon. The rest are going to be dendrites. You may or may not know which is the axon. So again, if you look at this and compare the dendrites and the axons, you can see that these are challenging to differentiate in this kind of a view and this kind of a stain. There are tricks I can use to very easily determine which of these is the axon. Okay. In terms of APR and identifying things, if you see one process highlighted, I want you to know axon. Okay? So if you see a whole bunch of processes come off, I'm only highlighting one, that's going to be the axon. On the other hand, if we highlight multiple, right, you know a cell only has one axon. So if I'm showing you multiple, those have to be dendrites. Okay? The cell body is called the soma. You know about the nucleus, I hope. Within the cell, we're going to have something called Nissl substance. Nissl substance, go back and refresh your memory if you don't remember what that is. You need to know not only what it looks like, but you need to know what it is. When we're talking about the nervous system, this Nissl substance is incredibly important. When we're studying structure, most of the human tissue has been analyzed using a Nissl stain. So it's something called cressyl violet or thionin, methylene blue. All of these are going to bind to and stain Nissl substance blue. So when you look under a microscope, you're going to be able to see the cell bodies really easily because you're detecting the Nissl substance. So if I want to count number of neurons, for example, Nissl stain is a gold standard. Now when we talk about the axon, there's a special region called the axon hillock. You may have heard it called the trigger zone. I'm going to say axon hillock. That's what neuroscientists typically use. The axon hillock is part of the axon, but it's just the beginning of it. Right? And this is where the decision, where a passive physical change in the electrical polarity of the membrane, where a decision is made whether or not to fire an axon. Okay, so if this part of the axon is excited, we will get the generation of an action potential that'll be carried and conducted down the rest of the axon. Right? So you're going to hear me talk about the axon hillock as the site of integration or the decision point. Okay? All right. Now in a more schematic diagram, again, this is what a typical neuron looks like. One thing I would like to point out, this neuron, I can tell whether it's from the periphery or the central nervous system. If you've had any neurobiology or remember your intro bio, you might be able to also. I'll tell you that this is from the periphery. This is a peripheral nervous system. Now, how do I know that? The neuron, the structure, the shape, dendrites, things like that, don't look any different, really, when we're talking about a peripheral or a central nervous system. Right? They have dendrites, axon, hillock, axon, all of that. What is different, though, is the type of cell that makes this myelin sheath. So axons are covered in fat. It's an insulating cable, basically. In the central nervous system, a type of glia called an oligodendrocyte, and we'll get to that in a minute, but an oligodendrocyte is going to make this fat coating. In the peripheral nervous system, it's Schwann cells. So the fact that I've got an axon that's coated in myelin, and that here I have Schwann cells labeled, I know that this is from the peripheral nervous system. You should definitely know that too. One of the reasons I bring this up is in Buffalo we have a very high incidence of MS. We also have a very well-known group at UB, affiliated with UB, studying MS. MS is a disease where the immune system attacks and kills oligodendrocytes. So, will a peripheral nerve like this be affected by MS? Not directly, right? There's no oligodendrocyte here. If we had a disease that killed Schwann cells, this cell would be in trouble, right? But when we look at that coating of the axon made by oligodendrocytes, if my immune system kills oligodendrocytes, I lose those beads of myelin. Right? We can see that. So what? You'll find out. But it makes it more difficult for those action potentials to be transmitted to the next cell. All right. So again, the myelin sheath, I talked a little bit. It's that fatty protective wrapping around the axons. It's an insulator, and here we're not talking about heat. When we're talking about insulation, in this context, we're talking about basically electrical current. Right? So it keeps electrical current in the axon. It's also going to allow for something called saltatory conduction. This is a very quick transmission of the signal. In humans, almost all of our axons are myelinated. Almost all of them have myelin. There are a few, notably the pain fibers, that don't have myelin. Other species, though, especially invertebrate species, don't have myelin. And so there's a lot of functional consequences and differences in how the nervous system works, how quickly it works between vertebrates and invertebrates, because some of us have this myelin sheath, some don't. All right. Now with the myelin, you probably already know that it's like beads on a necklace. That's a good analogy. There's a little bit of space in between each bead. That's called the node of Ranvier. If you've read ahead, and I hope you read before you watch these video lectures, you know that we'll be talking about sodium-potassium ion channels. Sodium and potassium ion channels are only found at the nodes of Ranvier. You don't find them underneath the myelin. Okay, so that has important functional consequences also. Now, when we look at myelinated axons, these are going to be quicker. This is too much of a simplification. It's not due to the nodes of Ranvier. It's due to the whole entire structure. Okay, and we'll get into that a little bit. But they are faster than unmyelinated axons. Again, is this better or worse? It depends on what you want to do. Sometimes having your nervous system work real quickly is good. Other times it's not so good. And so it's specialized for what you want to do. All right, and I mentioned MS is going to be a disease of the myelin sheath. Here, it's only in the central nervous system. Now, why do you have a loss of muscle function? It has nothing to do with those peripheral nerves. The peripheral nerves are fine. But it has to do with messages getting from the part of your brain that says, okay, I want to move, to the right parts in the spinal cord, right, that control those motor neurons that are going to cause contraction of those muscles. So if those signals aren't getting through steadily, I'm going to have difficulty with contraction. I'll talk a little bit about MS in a bit. All right. So when we look at the peripheral nervous system, again, when you look up here at this, it's kind of difficult to tell what's what, even if I told you this was nervous tissue in the peripheral nervous system. So for here, don't worry about identifying this on part 1. You still need to know the information, though, for part 2, and this is a great way to illustrate that. So here I've got my axon. Okay, so the actual axon, the part that's carrying that electrical current, is this. Right? It's that line. It's like a little tube. Here you can see where we might get a little space in between two separate myelin beads. And that's going to be the node of Ranvier. Now, each myelin bead in the peripheral nervous system is made by one Schwann cell. This is going to be different than what we'll see in the central nervous system. So if I kill one Schwann cell, I'm only going to lose one bead along the axon. And so if the Schwann cell that made this dies, I only lose this. This will be unaffected. No other myelin bead anywhere will be affected. I'm going to show you now what happens in an unmyelinated axon. Right? We're skipping ahead a little bit. The action potential is that signal that is going to go from my neuron to somewhere else, okay, be it a muscle, gland, whatever. So if my brain is saying contract and say this is a motor neuron, right, so my brain is going to send a signal that's going to excite this motor neuron. The motor neuron is going to generate an electrical signal, an action potential, that's going to go to the end of the axon, cause the release of, you should know this, acetylcholine, right? That acetylcholine is going to bind to nicotinic acetylcholine receptors. It's going to cause the influx of sodium, right? I'm going to get depolarization of the sarcolemma. When that happens, the sarcoplasmic reticulum is going to release calcium, right? And I'm going to get muscular contraction. Okay. So again, I've got to decide this neuron has to be excited enough to send that signal to the muscle that basically says contract. So here with an unmyelinated axon, I'm going to show you how we get from one to another.
During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
Okay, so what you can see in this unmyelinated axon, they use some words we haven't really talked about. Again, hopefully you read the chapter, though, and you have been at least familiar with those words. All right, now, when I talked about polarity, remember I said information gets collected by the dendrites, then sent somewhere. The reason along the axon that we go in one direction is going to have to do with the refractory period. That's going to be important to remember for later. Now, if you look at this unmyelinated axon, right, ions are going to flow in, they're going to diffuse to the side, they're going to depolarize the membrane, and I'll get the influx of new. And that's going to happen all the way down. If I've got myelinated axon, wherever there's myelin, I don't have any of those channels. I only have channels. I only get the movement of ions at the node of Ranvier. And again, because of the physical properties, to make a long story short, I'm going to get the influx of ions at the axon hillock, so it's starting my action potential. Now I'm going to get passive propagation of my signal very quickly to the next node. At that next node, ions are going to flow in, and that's going to very quickly move to the next node. Right? So in terms of opening of ion channels, the signal seems to jump from node to node to node. And when you do that, that's much faster than opening ion channels all the way along. You can think of it if you got a bunch of pool balls, right? So let's say you got 6 pool balls, and you put them in a line, and you leave a bunch of space in between each of them, right? You hit one, it rolls, hits the other, rolls, hits the other, rolls, right? That's going to be fairly slow. That's kind of like what's going on at the unmyelinated axon, right? That rolling would be the influx of ions. So roll, okay, roll, and we're going to get the transmission eventually at the end. But then imagine taking those 6 pool balls and just putting them right next to each other so they're touching. Now when I hit that first one, what happens? Yeah, the last one just comes shooting off almost instantly. Okay? So that's what happens with a myelinated axon. Okay? So that's a rough analogy as to why the myelinated axon is so much faster than the unmyelinated axon. Right. Now, again, what do these look like? Again, here's just a comparison. In the human, almost all your neurons, as I said, are going to be myelinated. Okay, so we're going to spend most of our time focused on myelinated axons. It's important to understand, though, that there are unmyelinated axons. These are going to be pain fibers. Right? So a lot of the pain fibers are unmyelinated. So you may recognize this if you've ever been unfortunate enough to put your hand on something that's real hot. Immediately, right, you pull your hand back. That's myelinated axons, monosynaptic reflexor. Okay, so it's very fast. Now, you've done some damage. It's going to hurt. Some, yeah, it may take a little while for those receptors to be stimulated. But even if they are stimulated, it takes longer for that pain signal to get to your brain than it does for the signals that are going to be essential for, hey, I moved. Okay? And that's because they're carried by different types of axons. The myelinated axons are much faster than unmyelinated axons. So again, to help you understand Schwann cells, the difference between myelinated and unmyelinated axons, right? In here, this is the axon. Essentially, what a Schwann cell does, here's my axon. My Schwann cell is going to come up, and then it's going to glom on. It's then going to take a little bit of its membrane and kind of start sending it out. Okay, so the whole cell doesn't move. It just shoots out a little bit of the membrane, and it's going to grow that membrane. And as it grows, it's going to circle around and around and around the axon. When it does that, we're going to end up getting a layered myelin sheath. Right? So it doesn't show up too well on this, but if you look closely, you will see that it's actually layers of fat, layers of membrane. And so we get a multi-layer wrapping. The Schwann cell, again, is this whole structure. The myelin sheath is part of the Schwann cell. You probably already figured out this is the nucleus. We need a nucleus to control what goes on. You can see that the Schwann cell is not much bigger than the nucleus. You can also see that this Schwann cell is not that big compared to the diameter of the axon. Most axons are pretty small. So if we were to draw a neuron, I mean, often you see it kind of drawn like this, right? Oops. That's pathetic. But you see something like this, all right? So we've got this big cell body. It looks like a swordfish, doesn't it? All right, I'm going to be simple. There. So often you see it drawn something like this. Okay, so we've got this big cell body, and then we've got this little process coming off of it. And if you looked back on the first pictures I showed you with the histology, you could see that the axon was very small. All right. So compared to a neuron, an axon is very thin. How big is a Schwann cell compared to a neuron? Now, it's a bit bigger than an axon is in diameter, but that's pretty darn small, right? So when we're talking about Schwann cells, we are talking about tiny little cells. Right, now compare this with an unmyelinated axon. Okay. Again, you're going to be able to see similar structures, right? So here are my myelinated axons. Here are unmyelinated axons. And so you can see, again, how they look differently. What's inside looks the same. The axon's the axon. It's got the same structures. Right? What's different, again, is the presence or absence of that fat or surrounding it. In the way that these are stained, the fat shows up as dark. So on this picture, again, here is my myelinated axon, and I can see, right, this big layer of black. That's the fat. When I look at the inside, though, right, these two things really don't look that different. Makes sense. They're both axons. They both have to do the same thing. Just one's myelinated, the other's not. All right. Now, I talked a little bit about the action potential. In a few minutes, I will go through, actually in the next lecture, I'll go through the molecular basis of the action potential. I hope you've heard it before. If not, give yourself a little time to pick it up. It's probably conceptually one of the more difficult concepts, not only in this unit, but probably the whole class. So make sure you give yourself enough time to know that. You absolutely will have to know that. All right. So when we talk about this electrical signal, basically the action potential is changes in the flow of ions. Positive charges are going to come into the cell, and that's how we get our signal. Right? Again, it's an electrical impulse. Okay? This shows you what happened in the video, that we're going to get positive charges coming into the axon. Those are going to leak a little bit to the side. Now, when they leak to the side, they're going to cause special channels to open up. Those channels are going to allow these positive ions on the outside to come in. Okay? So in an unmyelinated axon like this, right, channels are going to open. I'm going to get the leak to the side. That leak to the side is then going to lead to those ions coming in. Now, when these ions come in, what do? Yeah, they leak to the side. So when they leak to the side, what are they going to do? Stimulate these channels to open up. So then what happens? Exactly. The positive charges are going to come in. Okay? And so by this change, we're going to have the appearance of movement down the axon. Okay? Now with a myelinated axon, we're going to get saltatory conduction. And this is going to be the appearance of the signal jumping from node to node. Now, one thing they don't show particularly well in this diagram, there's only ion channels at the nodes. So I said when the charge leaks to the side, right, it causes channels to open up and ions can flow through. In an unmyelinated axon, you've got those channels all the way down the axon. In a myelinated axon, you only have them at the node of Ranvier. So if I were to ask, think about the membrane right here, where I just highlighted, are there ion channels there? No, there are not, right? How about here then, at this first node, are there ion channels? Yes. All right. So when these charges leak to the side here, are we going to get the influx of ions underneath the myelin? And the answer is no. Why not? There's no channels. There's no channels to open. Remember, the membrane is semi-permeable. So if I have charged particles, even if they want to flow because of concentration differences, they can't pass through that membrane. I need to open a special channel. Okay? So no channels, no flow of ions. No channels, no flow. Channels, I get the flow. Okay? So that's what's going to happen. Instead of leaking to the side and stimulating channels right next to it, leaking to the side, right next to it, right next to it, we're going to get a whole bunch coming in, and they're going to kind of side to this next one. Now they're going to cause these to open. These are going to come in and leak down to the side enough to give me opening of those channels. Okay? All right. We'll go into a bit more detail about this later. Again, if you read the chapter, and I hope you did, this is going to make sense. I'm going to talk quickly about different types of neurons and types of glia, and then that'll be the end of this little section here. So types of neurons, essentially what you do is you find the soma. Here's my nucleus. And then you count the number of processes coming off of it. Okay? So if I see one process coming off of it, one uni. Okay, so if I have just one coming off, I'm going to have a unipolar neuron. Most of the time, people just call them unipolar neurons. The sort of official name is a pseudo-unipolar neuron, pseudo-unipolar. I want you to remember, sensory neurons are unipolar neurons. Okay? We'll talk about these again in a minute. But if I say a neuron is a sensory neuron, you should automatically know that it's going to be unipolar. Bipolar, hey, I've got two, right? Two. Bipolar. Now, in this case, you know neurons have one axon, right? So if there's two processes, one is going to be the axon, the other is going to be the dendrite. Okay? Bipolar, there's a few examples of those. When we're talking about sensation, we talk about bipolar cells in the retina. You may remember those. We're also going to talk about bipolar cells in the olfactory epithelium. So there's a few places where we see bipolar. Right? So again, the easiest way to remember whether a neuron is pseudo-unipolar, multipolar, or bipolar. Pseudo-unipolar, remember sensory neurons. Just memorize it. Bipolar, remember the eye and the nasal epithelium. Everything else is going to be multipolar. All right, so that means more than two processes. So if we got 3, that's a multipolar neuron. If I've got 100, I won't draw them all. If I've got 100, multipolar. Most of the neurons in the central nervous system are multipolar. All right, again with the multipolar neurons, here's going to be an example of a multipolar neuron. This is a type of stain I want you to learn how to recognize. This is called Golgi stain. Golgi stain. You'll see a lot of Golgi if you do anything in neuroscience. Probably more pictures than actually doing it. The technique is a monumental pain in the butt. However, with Golgi stain, it completely fills a neuron. So if I stain with a Nissl substance, what am I going to see? Or sorry, if I stain with a Nissl stain, I'm just going to see Nissl substance. Right? So basically I'll see the soma real well. I won't see anything else very, very well. What Golgi does is it completely fills the neuron. And so I can see everything. If I'm interested in categorizing neurons by their shape, that's a great thing. Now, if you think about the brain, even with rudimentary knowledge, you probably realize that your brain is just packed full of neurons. And so if I were to take a piece of cortex and I were to completely fill every single neuron, what do you think you'd see? You'd see this mass of black dye. You wouldn't be able to see individual cells. There's so many and they're so tightly packed, it'd be really hard to see anything. Golgi, for some reason, and again, this is magic. I'm not really exaggerating. Only about 1% of neurons are stained. Okay, so here we're showing one neuron, one multipolar neuron. There's actually well over 100 in this view. Okay? Only about 1% of the neurons are stained. We have no idea why. We have no idea why it's only neurons. Okay? But it is. And we can take advantage of that, trying to classify things based on what they look like. Now here, when we've got everything filled in, right, strictly speaking, you can't tell the difference between an axon and a dendrite. Right? It's filled in. The structural differences in the cytoskeletal proteins that you can use to differentiate dendrites from axons are filled in. Okay, so you really can't tell. APR is identifying this as an axon. There's other ways in context you can make a guess as to whether it's an axon or not. As far as from a real neuroscientific perspective, though, that's a guess. You can't tell using Golgi stain. Okay, that's important. Now, the cell body, very distinctive. Most of what you're going to see, these processes coming off, are clearly going to be dendrites. Okay, in terms of which is a dendrite or an axon, in my class, we don't know using Golgi stain. Okay? What you can say, though, is these are all processes. And what you can say is one of them, and only one of them, is an axon. Okay. So again, what do these look like? Multipolar, bipolar, pseudo-unipolar. There you go. All right, summarizing for you. Now, I mentioned the glia. These provide support. There's a 10-to-1 ratio about of glia to neurons. Neurons divide very slowly. All right. We used to teach, people used to think maybe 10 or 15 years ago that you're born with all the neurons you'll ever have. You don't produce more as an adult. That's not true. In the hippocampus and olfactory bulb, there seems to be neurogenesis in the adult. Okay, so the adult is going to make more neurons. Now, it's not very many, and clearly they're not very good at compensating for mass damage. And so neurons really, for all practical purposes, don't divide. Okay, so neurons don't divide. Glial cells do, though. You can make tons and tons and tons of glia. Now, this actually has two implications. Cancer is essentially a lack of regulation of the cell cycle. So cells begin to divide when they shouldn't. They stop following the rules, so to speak. Neurons, which aren't dividing, right? They're not dividing. They're already fully mature. You think these can form cancer tumors? They're not dividing. They're not even playing the game, right? They don't need to follow the rules. So it turns out neurons don't form cancer tumors. There's no such thing as a neuron cancer tumor. The glia, however, are dividing very, very rapidly. There's the opportunity for pathology. Okay, so because neurons don't divide, glia do, we can have glial-based cancers in your brain, but you don't have neuron-based. Okay? All right. Now, the book's going to talk about 5 different types of glia. Astrocytes are cells that are kind of star-shaped. Right? Here's an example of an astrocyte. Astrocytes have a couple very important functions. One is they wrap around the blood vessels and the capillaries that are in your brain. These help form what's called the blood-brain barrier. So neurons are incredibly picky. Once a neuron is dead, it's gone forever. You're not going to replace it. So you need to protect those very, very well. The chemical environment is very tightly regulated. One of the ways they do that is things can flow in this capillary, right? They're in your general circulation. However, these endothelial cells are specialized. Okay? Normally in capillaries, there's little spaces between the endothelial cells, so substances can easily diffuse in and out of the capillaries. In the brain, there are no spaces. So in order for a substance to get in or out of your brain, it's got to go through one of those endothelial cells. So that's one level of regulation. Once it makes it through the endothelial cell, we still have to deal with the astrocyte. Okay, so the astrocyte is another layer of protection. If a toxic substance, for example, makes it through that endothelial cell, the astrocyte has a chance to grab it and keep it from getting to a neuron. Okay? Again, you'd rather kill an astrocyte, because I can make more, than I would a neuron, because once it's gone, it's gone. When we look at glia, again, I told you it's already 10 to 1, right? Glia to neurons. Of the glia, though, the most abundant form is the astrocyte. Another thing that the astrocytes do is they regulate the synaptic environment. If you've read, you know what a synapse is. Okay. If you haven't read, again, that's why you should stop this and go read. But the astrocytes help regulate the synaptic environment. All right. There's other glia called ependymal cells. I want you to know the word choroid plexus. Choroid plexus. This is a term for the cells that line the third ventricles. We'll get to what the ventricles are. Sorry. They line the ventricles, not just the third ventricle. Okay. There's cells that line the ventricles. These cells, these ependymal cells that form the choroid plexus, make cerebrospinal fluid, CSF. We'll talk about the importance of that a little bit later. But choroid plexus is a term that neuroscientists use very commonly. So I just want you to be aware of that word. All right, we're going to have microglia. Micro means small, glia means glia. So these are going to be very tiny little glial cells. And again, in relation to a neuron, you know, a neuron is going to be maybe a semi-trailer. A microglia will be your little Vespa or something. All right, there's a huge size differential between the microglia and the neurons. The function of the microglia, though, is to help clean up. Okay? So they help remove bacteria, viruses. When cells die, when there's damage, these microglia come in and help get rid of cell debris. So these are important for helping prevent infection and dealing with brain injury. In fact, there's a term called gliosis. In animals, we see this a lot. In humans, hopefully you don't get to see it. But if there's damage to a part of the brain, you slice the brain real thin, stain it for a Nissl stain. These will show up. all cells, neurons, glia, whatever, will show up using a Nissl stain. But what you'll be able to see is very dark, dense dots all around where there was damage. And that's because these glial cells came in to try to clean up the waste. I mentioned the oligodendrocytes, okay, these myelin sheath in the central nervous system. Now, one thing to notice, one oligodendrocyte, okay, this is all one cell, is going to send out feet. Each foot is going to form one myelin bead. And these can be on different axons. They may be on the same axon. They may be right next to each other. Okay? They can be kind of all over the place. Oligo means a few, like an oligarchy. Dendro has to do with feet. Cyte is a cell. Okay? So these are cells with a few feet. Each foot, one myelin bead. So when I kill this one cell, I don't just lose one myelin bead. I lose many myelin beads. MS, that's exactly what happens. The immune system seems to attack these oligodendrocytes. Now we've got some bare myelin, some bare axons. We don't get efficient transmission, and that can lead to some problems. Now, I mentioned Schwann cells, so I'm not going to spend any time on those. We've already talked a little bit about those. Notice, though, how we get the wrapping around. All right. Here's a great summary. With the neurons, make sure you know the different types. I want you to know pseudounipolar are sensory, bipolar. These we find in the retina and the olfactory epithelium. Everything else, multipolar. For the neuroglia, I want you to know what each is, be able to recognize a cartoon picture of it. So one of these, I'm not going to ask you to identify it on APR or anything like that, but have some idea of what it looks like. But most importantly, functions. I'm going to pause here, and when we come back, we're going to start to get in